Good morning, everyone. Welcome to Kemin's Masterclass series this morning. I'm Eileen Han, Head of Animal Nutrition Platform at Kemin Asia. I'm once again MC of the day. It's a golden rule of animal production is that healthy animals are more likely to provide higher productivity and more economic values. In this sense, immunity priming or immune modulation is an interesting concept that can be introduced to our industry and is about optimal level of activation of immune cells to get them ready for fighting against pathogen and antigen in immune system. Professor Chul-hee Yoon from Seoul National University, the speaker of the day, is a renowned immunologist in Korea who has also animal science background, will walk us through from basic immunology to specific topics in immune modulation to give us solution to figure out the best option in this era of infectious diseases. So at the end of the presentation, there will be Q&A session based on the question you will raise at the chat box at the bottom. During the presentation or at the end of the presentation. Please feel free to raise your question by typing in chat box. Then we will select the most frequently asked question for the discussion with the professor. So without further ado, let me invite Professor Yoon. Please join me and welcome Professor Chul-hee Yoon.
Well, thank you, Eileen, for a nice introduction. Can everybody hear me okay? I guess it's okay. And good morning, everybody, or maybe good afternoon, or even good evening in some part of the world. Although this is not the way that I would like to interact with people, obviously COVID-19 definitely changed our lives. Thus, welcome to the webinar. I am Chul-hee Yoon at Seoul National University. I would like to talk today about immunomodulant. As Eileen suggested, if you have any question during the lecture, and you can leave your question on the chat. And also I'll have some email address later on that you can send me an email as well. So just briefly, one slide explaining where I am, although you all know already. I'm here in Seoul, Seoul National University in Seoul. And if you have a chance to come to the campus, then this is what you will see during the night. And we do animal immunology. What I would like to do today is that a very brief introduction, and would like to discuss a little bit on major issues in domestic animals in relation to the topic today. And very brief, few slides explaining about immune responses, followed by immunomodulant and beta-glucan a bit, and conclusion. And between in here and at the end, we'll have a poll time so that we can briefly go over with what I'm saying today. Just for your information, I'll let you know. So if we look around animal farming, we can easily notice that there are climate change issues with global warming. And also it's been a while with the issue with AGP, antibiotic growth promoter. I'll touch this one briefly as well as a major issues. And believe it or not, Korea was the first country in Asia to ban the use of the antibiotic in animal feed. But at the same time, we all are aiming at high productivity. Well, there's more. We have a situation with a pandemic disease. Well, the reason we do this way, the webinar, is obviously because of this coronavirus, the lately COVID-19, but we have had this one already a while ago, SARS and MERS. They are all from coronavirus. And in fact, they were with other primary host that is now jump onto us and causing the problem. Well, it's not only human, but also we've been struggling with the disease on domestic animals, FMD, avian influenza, and lately ASF. And I write up the editorials in AJAS. If you look at the incident or outbreak, at the end of the year 2018, there was a problem in China that quickly spread to adjacent countries, Mongolia, Laos, North Korea, Hong Kong, Vietnam, South Korea, and Philippines, and all that within few months. So the impact of disease is enormous these days. And we have problem. If I put all this together, including previous slide, I would say there are various psychological and physical stressful conditions on animal farming. So what's the stress? It's a response to a stressor, and stressor is a root of stress. It could be an agent, but also a condition or an environment. It could be anything. Well, believe it or not, there are negative part of stress, but also positive side of the stress as well, and both mental and physical well-being. If you look human first, and then I'll show you the domestic animals in the next slide. But briefly, there are positive side of body function and energy, motivation and relationship and controllable life. That will be positive side. But oftentimes we'll see negative side in terms of anywhere from high blood pressure, that's physical effect, irritability and sleeping problems, that's emotional, and memory problem and depression, that's mental effect. Now, if you look at this in domestic animals, we can categorize into a few. Nutritional factors, unbalanced nutrient, toxin, starvation, diet changes, and also environmental factors, that's a temperature problem and microbiota, and some others. There are biological factors, weaning, infection, vaccination, and castration and trauma, but also physical factors, including transportation and social status for the domestic animals. Some of highlighted in here with the red is something that we have done in our lab for past many years. Well, I don't think we have much time to go through, but I'll introduce briefly later on. So in here, I would like to spend a bit of time looking at major issues in domestic animals. Especially in relation to the topic today, I thought of 4 different parameters or agenda: antibiotic and antibiotic replacement, and anti-inflammatory reagent, and appropriate symbiotics, and vaccine. As I mentioned already, that Korea was the first country to ban the use of antibiotic as a feed additive, followed by some other countries, including Vietnam and many Asian countries. And even China announced a while ago that they will do this year. And we will see what happens with this announcement. And with antibiotic use as a feed additive, I recommended to this opinion article published in Frontiers in Microbiology. It's only 2 pages, so it will not be that difficult to go through. And key point in here is that they say 2 things, actually, which we already know. One hand, yes, it's true that we have had about 10% of gross promotion. At the same time, if we look back past many years, there has been increase of antibiotic resistance And that's the fact. So it's saying that there are two things and trade-off with increase of antibiotic resistance. And therefore, people are looking for alternatives to antibiotics. There are two different way of approach these alternatives. One is growth promotion. The other one is for disease prevention. If you look at the growth promotion first, one can use feed enzymes, probiotics, prebiotics, or symbiotics, and AMP or organic acid. That could be alternatives. I will not go through details of each of these because of the time constriction. And then we look at the disease prevention. Well, definitely vaccine would be the first choice because the prevention is always better than treatment. And especially for domestic animals, we are reluctant to do this treatment. And immunomodulant, that's the topic today. So I think I will touch this one a little more later on. So I'll save this for now. But also there are increased use of bacteriophages and endolysin because they seem to have a very specific action against pathogens that has been known already. And although, also there are other disease prevention alternatives. And I cannot emphasize more on sanitation over any other parameters that I'm talking. Of course, sanitation first, based on farm management and biosecurity program, that would be top priority. But still, we know that sanitation, doesn't matter how much you put your effort, still we may need some of these, and I'll come back to that later. So what's the issues in prospecting? Well, we know that we have some antibiotic residues problem and antibiotic resistance problem, and especially there's a lacking of a next generation antibiotics. Well, I think we know quite a bit, but still I question myself that do we know enough in terms of mode of action mechanism, status of microbiota, and host immune responses? And I think that we are still in need of studies on antibiotic replacement for growth performance and disease resistance. So what I suggest is that we may look for some relationship among antibiotics and nutrition, microbiota, and immune responses in order to find the proper antibiotic replacement. Well, second, anti-inflammatory reagent. I always emphasize, although we regard human and mouse and domestic animals, the system would be similar in terms of immune response. They're not the same. Well, in human, we look a lot more on cancer and autoimmune disease and et cetera, et cetera. In domestic animals, we do not. And also, one must think carefully when domestic animals get benefit from using anti-inflammatory reagent, so cells and molecules to growth performance, especially, you have to think inflammatory responses are not always essential, especially for the vaccination. Well, probiotics and prebiotics and symbiotics, it is all about the microbiota and host. Although prebiotic is the food for the probiotics, but still, eventually at the end of the day, we're looking at interaction between microbiota and host immune cells. And each, meaning that each strain of probiotic would have different role as a growth promoter and also to the host immune response. And the vaccine will be traditional vaccine, but there are vaccines that are advanced or newly suggested. In general, we use whole microbes as an inactive form. And DNA, subunit, conjugated, or synthetic vaccines are coming after. And a lot of companies and research units are putting their effort to find some new way of doing this. And also the route and dose and type of the vaccine would be a key question. And also the adjuvant cannot be separated from the vaccine when we talk about the efficacy. And especially when you're looking at all these disease that has been problem for us in human and domestic animals, they are mostly coming into our body through airway, that's a respiratory tract, or intestinal digestive tract. And therefore, perhaps we may need mucosal vaccine rather than systemic vaccine together with the proper adjuvant. I will not go much details on this part neither, but I thought that this would be a good time to think and consider together with the immunomodulant. And for the disease control, what should we do? Well, I think there are different things has to be done in each sector, and the field, government, and research unit. On one hand, in the field, I already emphasized the importance of the sanitation and vaccination, but also we have to follow the rules, like active alarming at disease occurrence, for instance, so that we can minimize the damage, not only to myself, but to neighbors and maybe countries, if you'd like. And also for the government, they have to set the proper policy and regulation. Oftentimes I see, even in Korea, that there is a policy and regulation go way behind, or sometimes goes too fast, early enough that no one else is understanding, and that's the problem. Has to fit with the other unit, field and university. And also should have a long-term plan and monitoring platform and support. And support part, I'll come back with this one later on, how we can do it at the end. And also in the research unit, well, research unit is not the company. We're not making product, but we should study and help the development of other product for the company, for the field. Well, with that, I think the first part of my talk is done. And this is my email address. And for now, we'll have poll time. Eileen?
Hello. This is the first poll. And then
Louder, please.
hello. Hello. Can you hear me clearly?
Yeah. Now it's
okay.
good.
Okay. This is the first poll, and then going to be very brief. Okay, I will launch the poll here. Okay, hold on a sec. Okay, so this is the question. So which of the following AGP, antibiotic growth promoter replacement, is your current choice in your diet? You can choose multiple choices.
Oh, it's not easy one.
There is no right or wrong answer. So you can just, we try to understand the trend. Oh, okay, still keep coming. So let me wait another 20 seconds.
Sure.
Okay. Okay, maybe then maybe I can stop here. We cannot wait forever. So, okay. So here is the result. Can you see the result? So
Oh, yes.
winner is probiotics.
Probiotics. Okay.
Probiotics is widely used in our industry, followed by organic acid. Then phytogenic and immunomodulant is a similar percentage. So, and then the interestingly, the people who answer do not use anything, the none is only 1%. So meaning mostly we are using one of the AGP replacement in the diet. So this is very interesting session. So back to you, Professor Yoon.
Sure,
okay.
Sounds good. Well, I learned something as well, probiotics, wow. Well, there is some movement from probiotics to propri and symbiotics, and we'll see how it's gonna be changed in the near future. But thanks, Eileen. I'll go ahead with the next chapter. It's about the immune responses. I know you already have headache, but I'll be brief, I promise. So the basic concept of immune response is about the self and non-self. For example, cancer, that should be non-self, and immune system should attack those cancer, yet they do not. And they were actually self at the beginning. They were just one of those cells in our body. And all of a sudden they change as abnormal cells. But immune system is not recognizing. So for us as an immunologist, as a medical doctor, our goal is to make immune system to recognize those cancer as a non-self so that they can attack. Pathogen is the same thing. We have to make immune system recognize them early enough so that we do not get much damage. So there are three things in order, and that explains immune response. First one is recognition. Anything and everything we talk in immunology is about the recognition. Because without recognition, there's no such a thing as a specificity. So in order to have a specificity, there has to be a recognition. I'll come back to that briefly later on by using specific molecule or how they interact, including beta-glucan. When response, well, once they have this interaction, they are changing. The cells are changing. As they change, they produce some proteins, immune proteins that we say effective response. But at the end of the day, once they have this interaction and produce some response, effective functions, and this is the key core of the vaccination. The reason we vaccinate ourselves and domestic animals, because after the vaccination, they will have immunological memory. That's the key. So these are the basic component of immune responses. I tried to draw or brought some schematic diagram to make you understand better. There are, let's say this is the time after the infection, okay? And from hours to days. There are two arms of immune response. One is innate immunity. Okay? One is innate immunity. The other one is adaptive immunity. Innate immunity, well, there are commensal microbiota, but also some pathogens, and there are front line, a first line of defense as an epithelial barrier. And underneath, there are immune cells together with a complement. That we call the composition of innate immunity. Well, they are good at immediate response. They act right away, but they don't have any specificity. They don't have memory. Whereas adaptive immune system, we talk about B cells and T cells. And we say a lot about the antibodies. Antibodies, the B cells are producing antibodies. B cells are only cells that produce antibodies. And there are T cells, also a part of adaptive immunity. And adaptive immunity, compared to innate, they are relatively slow. It takes a few days to get there. But they are highly specific and very diverse. And also, especially, they do have memory response. if I put this thing together, there are two important events. First of all, that has to be recognized somehow. Okay, and then those information has to be transferred to adaptive immunity for the specificity. In other words, adaptive immunity can start only when there is an innate immunity. So innate immunity, there will be some inflammation, and also importantly, there's a tissue repair once there is a damage. So innate immunity is essentially required for effective responses of adaptive immune system. And thereafter, once we have, as I said, when there is innate immunity, and that adaptive immunity can start. And cytotoxicity and T-cell differentiation, you don't have to care too much about it, but innate immunity important for the adaptive immunity as well. And most of the time, we are dealing with food and feed ingredient, and those are going through gastrointestinal tract, that this is the some key factors. Epithelium, they are meeting with a huge burden of antigens, foreign antigens, including food antigens, microorganisms, and toxic molecules. Some of them are harmful, some of them are we must have. So there are definitely mixed antigens. And as we know, the primary function of the epithelial cells are for digestion and absorption of nutrient. It's key. But not only that, they do have a crosstalk between these immune cells and epithelium. As I said, they will be underneath of this epithelium and they have interaction. And also, they have tight junction proteins, which will not allow any pathogen to go through. And as you have more of this protein expression, that will be better for epithelial cells for the protection. Autophagy, well, I'll not go there much, but that's also one of the protection mechanism. For us as a human, we use these 5 senses to recognize it. But for immune system, and they have to have receptors to recognize, but also to communicate. So when there is a foreign materials, let's say pathogen and damaged cells, that has to be recognized by receptors. And also for the communication, they should have ligand and receptor, also these soluble factors, immune proteins produced by immune cell, also act on orocrine and paracrine manner. So those molecules are called PAM and PRR. PAM stands for pathogen associated molecular pattern, and PRR stands for pattern recognition receptors. I will not go through each and every molecule in this schematic diagram that I'm going to show you right away, but I want you to know that there are PAMs from microbiota and non-self other than our body. And our immune cells would have those receptors to recognize them. Many of them will be on the cell surface, but some of them will be even inside the cells. There's a reason for that. Viruses normally expose themselves in the cells, not outside the cells. And therefore, all those nucleotides that viruses would have will be recognized by those receptors inside the cells. That makes sense, right? Okay. And once they do, then they have these all different types of signals, and at the end of the day, we'll have immune response. Well, let's quickly move on to immunomodulant, the topic of the day. before that, I would like to emphasize domestic animal immunology. Why we need that? Well, there are so many different scientific background behind this, but I always emphasize mechanism and function, or the overall and scheme of this immune response and immunology would be similar between human and mouse to domestic animals. But if you look into details, they are not the same. And therefore, we should know exact mechanism and function. Why? Well, that will improve our understanding of the basics in terms of immune activation and tolerance. And also, what we need, even in the field, companies, for the development of mechanism-based product. It's not just showing increase or decrease body weight, let's say. Then you have to come back with others. And then you again do the same thing. Rather than doing the observation-based studies, I would like to suggest looking at mechanism and function for antibiotic replacement or vaccines and adjuvant and anything related to domestic animals. Therefore, we can have enhanced productivity of domestic animals. Remember I said these are alternatives to antibiotic for disease prevention, and one of them, well, all are important, but one of them was immunomodulant, and I promised you that I'll come back to this one and we'll have a look at the immunomodulant. So immunomodulant is the agent that modify the relationship between non-self, for instance, invading organisms or pathogen, and the host by enhancing host immune responses. And there are many terms that used together with immunomodulant: immunostimulant, immunoenhancer, immune regulator, immunorestorator, BRM, that's biological response modifier, and immune augmenter. Some are slightly different from one to the other, but they are all synonym. Why use the immunomodulant? Well, there are two different parts, but I'll not go a lot of details, but I'll show you some evidence later on. Activation of general innate immune response, but also for the adaptive immune response as well. For innate, to induce proper immune response against infection, and also to maintain enhanced general protection. For the adaptive immune response is to achieve the best outcome of vaccination, for instance. So by using this immunomodulant, you could achieve the enhancement of innate immune response, but also at the same time, you may have a benefit for adaptive immune response as well. And there are some immunomodulating polysaccharide, many of which have been studied for past many years. Beta-glucan from yeast, mushroom, cereal, that's oat and barley, and also bacteria and algae. And also there are other than beta-glucan as well, dextran and aspergillus, for instance. Well, as I mentioned, it's all about the ligand and receptor, recognition first. Before we discuss anything beyond, that has to be recognized. Very early studies, 1950s, we didn't know the target, but now we know many targets and interact with those immunomodulants. For instance, Dactin-1, toll-like receptors, and CR3. And how they do that, I'll come back to that point briefly later on. And target cells are mostly macrophages and T cells. Why? Well, simply because those cells expressed these receptors the most. And also, other than polysaccharide, there are oligosaccharide and from aloe and chitin and chemical compound as well. So there are some other choices as well. And if you look at the mode of action, There are direct activation and indirect activation. We should realize that it's not just direct activation, but also there are indirect activation. Direct activation would be, as you see in this schematic diagram, there's a regulation of barrier function to the epithelial cells. And also recognition by immune cells, dendritic cells, and macrophages. But perhaps more importantly, indirect activation through epithelial cells together with microbiota. And recently, there's a metabolite produced by microbiota has a quite important role on not only a ruminant, as we know, for the short chain fatty acid, but still for the monogastric or non-ruminant animals as well. In order to understand all this, I think it is important to understand gastrointestinal immunity, which we already know some of those are gastrointestinal tract development, digestion and absorption, and growth performance. But also something that we should consider is that mucosal immune system and disease protection. And therefore, I would like to categorize this into three main functions of immunomodulant: regulation of intestinal barrier function, and promoting regulatory function in the gut, and enhancing innate immune responses. So first, regulation of intestinal barrier function. There are a few things that under consideration. There's paracellular permeability. And okay, there's questions coming on. I'll have a look at it later after this session. And we have choices for immunomodulant. And we have done some studies in our lab. They're looking at the tight junction proteins. And when you have toxin, that's a decreased, while you use the immunomodulant and there are signalings and gives better protection of it. And that has been published a while ago. And also there are AMP and also protection against pathogen infectivity. And we have looked at the impact of this immunomodulant and together with a receptor that recognize it and AMPs that has been published. And also when you use the compound that also induces autophagy that protect PDB infection, that has been published as well. Well, regulatory function is the other side of immunomodulant. And when people used metabolite and some of these immunomodulant, and they found all this recognition system that induces production of chemokines and cytokines, which induced activation of immune cells, but also recruiting immune cells to the site. And therefore, at the end, you will see this immune cell recruitment and cell differentiation, and also increase of antibodies, for instance. We have done some of those studies as well in the lab. And we have used this immunomodulant and looking at the factors to have interaction between epithelial cells to immune cells. And that has been shown in systemic immune system, but also intestinal immune system. Interestingly, we have used atopic dermatitis, and that symptom is on the skin, not the intestine, but it was interesting enough that for us to see the reduced inflammation, looking at some other factors that are not described heavily in here. That's been published this year. And on the other end, there are enhancing innate immune responses. There'll be a number of choices: peptidoglycan, flagellin, and beta-glucan, and they all now, the receptor is well known, NOD and TLR5 and dactin, for instance. And even the mechanism of this compound, immunomodulant. Epigenetic reprogramming, and metabolic reprogramming, and also increase of responses. And also there's a term coined with this action as a trained immunity. I'll come back with trained immunity later on, but briefly, since you will have increase of this basal level of activation, eventually you will have better immunity as time goes by. And I'll come back to that point. And let's look at the beta-glucan. Well, if you look at the literature all the way back to the early 1950s, and you will find the term, say, zymosan. That's the in fact crude form of beta-glucan. And now we know that they are produced from yeast, algae, and cereal. Just for your information, they all have all this fungus, bacteria, yeast, and cereal. They have 1,3, beta-1,3 backbone with slight different side chains, beta-1,6 for fungus, bacteria, and yeast, and cereal for 1,4. And for your information, cellulose will be beta-1,4. Again, remember I emphasize the recognition and activation. Without recognition, you don't have activation. And I had my PhD on beta-glucan. By then, we didn't know the receptor. We assumed that there are a CR3, but we didn't know about the dactin-1. Now we know exactly the receptor for this beta-glucan. So there are two pathways. One is dactin-1 mediated pathway that alone or together with toll-like receptor, they induce the activation. And also there is a CR3, complementary receptor pathway. That's a CR3 mediated. At the end of the day, we will have cytokine production and B-cell T-cell activation and cytokine ROS and also immune cell migration. This is not what I'm saying, but if you look at the literature and you can find this interaction together with some of the outcome. I got this from information from Camin, and what it shows you is that beta-glucan is actively ingested by macrophages. Here is the control macrophages, and of course you don't see any of those beta-glucan, yet when you culture macrophage together with beta-glucan, now you will see beta-glucan, they are inside the macrophages. When you merge this shot, then you will see they are inside. This is the nucleus. And this is EM picture shows that indeed you have some of these part as a beta-glucan as well. Also, beta-glucan modulate a balanced cytokine by macrophages and dendritic cells. There are different parts of immune activities. One is activation, and the other one is inhibition, and also migration. So they are doing this in a dose-dependent manner. And this was from Camin as well. And also when they looking at the chicken with coccidiosis and control infected one had a lesion score high, whereas when you use the beta-glucan was reduced significantly. And the reason for this was probably you have infiltrated immune cells and macrophages at the site. And also beta-glucan seems to enhance the protection against the Newcastle virus. Of course, in this case, they do not use the virus, but they looking at the efficacy of the vaccine. And without vaccine, when you challenge the virus, and you will see this index would be 60% index by being herd immunity. And when you use the vaccine, that improved. But when you use those together with the glucan, that further increased to reach the point where we normally call the threshold for the herd immunity. You may have heard this one during this COVID incident during past few weeks or so in Sweden. Beta-glucan induced trained immunity. This was what I told you before. And since because of the changes, this was the beta-glucan papers published in Cell Report just a few weeks ago. And they seems to induce a rapid protective response. The reason because they were interactive with the gut microbiota that induces some initial response. That will do reprogramming here, epigenetic memory. And therefore later on, they will have better and best or quicker responses against other heterologous infection. They call the trained immunity. Well, just a few slides showing you what we have done in our lab. The lab is lab animal immunology. We do immunology, domestic animals, and infection and vaccination. We do nutrition immunology and vaccine and mucosal immunity and microbes. But more importantly, we looking at the function and mechanism. And during the past few years, we've done studies on pigs, produced some papers, and also chickens. We have done some studies, and these two actually got just accepted. And also cattle a while ago for stress response. And when you do domestic animals, we have to look into mouse and human studies because they have good fundamental answers. And therefore, we have done some immune cell regulation, mucosal immunology, and nanoparticle and vaccine studies by using mouse models and some of the human samples. So how to strengthen the impact of the immune regulation in domestic animals? That was the question at the beginning. And I think the balance is the key. Nutrition, immune response, and animal productivity. We know that when we provide the nutrition, they have the some impact on hormones and growth and immune responses. And in terms of the immune response, they should not too robust, but not too weak, neither. And also we know that there are quite complicated interaction among these parameters. And therefore the balance is the key, maybe immunomodulant. Then what should we do for that? And I told you already about this farm, field, and government and research unit. And I'll not go through again, but the government has an important role. And they should provide support to the research unit, but also to the field. And therefore we can work on mechanism and function so that we can provide those information to the company so that they can make a product to provide field. Important on national and international, and we should work together. Well, this may be a small step, but I'm sure these actions and effort taken at this very moment are for the future of next generation and for better sustainability. Take-home message would be target cells of immunomodulant is not just epithelium and immune cells, but interaction among those cells. And we can enhance gut immunity by using efficient immunomodulant. And still, I think we need a model to study interaction of aforementioned. And I'll not go through in here, but each unit has its own role to keep in mind that work together, especially the government, I say, make people happy, at least trying to. And I won't be here without help from the lab members and the collaborators. With that, and thank you very much. And now we have, this is my email address, another poll followed by Q&A. Eileen?
Okay, thank you, Professor. It was really informative and interesting presentation. A lot of scientific later part, but I hope that you all digested very well. Okay, here is the second poll. Okay, here we go. This is the second question. Which of the following production stage do you think the most immune suppressive?
Oh, this is
fun. All right. So once again, there is no right or wrong answer. So we try to understand what do you think when it comes to immune suppression in different production stage? Okay, so maybe we can wait another 10 seconds. Let's keep coming. Yeah. And then also we have a lot of questions still coming in. So shortly after this poll, we will address these questions one by one, but some redundant questions I will summarize as a one question. Yeah. Maybe I can stop here. Okay. And I will show you the result. Okay, so most of you think, the most immune suppressive the stage is definitely under stress condition. Stress meaning like heat stress, high density, or feed change, or different type of stress condition over here. And then followed by, of course, young animal. So young animals are vulnerable because immune system is not fully developed the inside of body. Also vaccination period is also stressful. And then the breeder, of course. So actually 4 of them all relevant. So yeah, thank you so much for your participation. So now we move on to Q&A. Okay, so we have actually many questions, but the first question, the relevant to everybody, and then this is the most frequently asked question. What is the difference between beta-glucan and prebiotics, and then MOS, mannan oligosaccharide, and all different type of, immune modulant?
Oh, sure, Eileen, but a number of people saying that they cannot see the poll. Can we quickly check, and you may work later to show those poll later?
Yeah, actually, the question
it's
lays
from separate window, right?
Yeah, it's a separate window. This question only shown to panelists and professors. So that's why, yeah, that's why I try to read the question out first.
Okay, okay, sure. Perhaps people will have interest in those results of the poll. So perhaps you can share later.
Oh, so you couldn't, they couldn't see the result of the poll?
Yeah, that's what some of people said in here in the chat.
Okay, then I will share once again. So how about right now?
Well, I can see now and before, but I'm not sure about others.
Yeah, but I understand that this type of technical problem can happen, I mean, on the individual computer. So maybe this is a specific problem just for them. So I can
maybe
see.
People confused a little bit.
All right. Okay. So shall we continue with the Q&A? So
sure,
as I said, the first question is what is the difference among the all different immune modulants such as beta-glucan, the MOS and prebiotics? Yeah.
Sure. There are some differences, and beta-glucan, I think I have shown you a number of different features. Mannan, for example, is the plant polysaccharide, and they have 1,4 linkages. Like oat, beta-glucan, and barley, they will have 1,4 linkages, 1,3 and 1,4. But mannan would be 1,4 linkage. And also, people may know a lot about probiotics and prebiotics, and I don't think I have to explain for that. And there are terms describe oligosaccharide and polysaccharide. For example, beta-glucan will be polysaccharide. Oligosaccharide will be number of monosaccharide, like a sugar, moiety, will be less than 10, no more than 10 monosaccharide unit will be oligosaccharide. And anything above, and normally beta-glucan is like many hundred, this glucan linked by 1,3 and 1,6, or 1,3 and 1,4 for a cereal. beta-glucan. So that's polysaccharide. Would that be enough? For if there is other questions, then I'll take it.
Yeah, actually, we can discuss this topic for 1 or 2 hours, I understand. So of course, actually, we will email them with a more detailed the answer we can provide to them by email separately. So let's move on to next question. Will a healthy bird benefit from immunomodulant, or will it be like a probiotic that can only affect or benefit poor performing bird? So basically, is immunomodulant also can be effective against healthy bird, healthy animal?
So question is whether the immune modulator would help or beneficial to healthy bird or a healthy animal. Definitely. One of the reasons we think the beneficial to use immunomodulant is not because of they sick, but thinking of a few things at least. One is, as I said, as a antibiotic replacement. I don't think they can replace, because I don't think they will get 10% gross promotion yet. We know that it doesn't matter how well we keep the sanitation, there are things that will be stressful to the animal. In that situation, we use immunomodulant in order to prevent or minimize those damages by stressful condition. And therefore, I think that it is more for the healthy animal than sick animal. Oh, I cannot hear you, Eileen. Yes, now I can.
Yeah. All right, that will be a good answer already. So let's move on to next question. Next question is, does an immunomodulant that is administered orally help in developing systemic and then laboratory immunity also, or only GI tract immunity?
Oh, yes, that's a good question. So there is a gray area between adjuvant and the immunomodulant. And when we say immunomodulant, in general, we think of enhanced baseline of immune system. But adjuvant, that's about the vaccination. And therefore, whether that will be beneficial to give this immunomodulant through the only orally or some other way. I would say if you use them some other way, then that has to be together with a vaccine maybe. And I don't think, well, first of all, it's a whole lot easier for us to do oral administration rather than other route, for sure. But if that is to be used together with vaccination, for instance, then you have to think about whether the efficacy of the vaccine would be better when you use this together. And therefore, I would say primarily, and you think this for oral administration, so that they will go as a feed additive, for instance. That will be the priority. And some other route, or you can think of some other route, but I think we need some more evidence, and the way you do has to be rather precise. Thank you.
Okay, thank you, Professor. Next one is, what are the differences between beta-glucan, 1,3 beta-glucan and other beta-glucan, such as 1,4 and 1,6 in terms of immunity?
Oh, yes. I think Eileen knows better than I guess, but it's a while ago when I have my PhD on this one, 20, 30, I don't know. I don't remember. But yes, definitely. I briefly explain about the beta-glucan in terms of the structure and linkage. And first of all this is about how monosaccharide is combined or linked. So if you look at the linkage, and they will have 1,3 backbone. And the way they connect to one to the other would be either 1,3 or a 1,4 or a 1,6. And therefore, there are some differences, but still, when they are recognized by Dactin-1, and that is about the 1,3, so you don't have to worry too much about side chains, yet they are not exactly the same among beta-glucan from the different sources. And some studies suggest that I believe that they have more of the beta-glucan than the other source. But more importantly, I told you that the oligosaccharide and polysaccharide, there are differences in terms of the how many monosaccharides are there to define as an oligosaccharide and to define as a polysaccharide. And therefore, during the process, if it's too harsh conditions, then you will lose this monosaccharide moiety, meaning that you will not have enough number of monosaccharide glucose linked together, or dextrin, depends on how you prefer. And therefore, when they say beta-glucan, then you have to look into some details. For example, even companies, they have beta-glucans from all different sources, and how well conserved as a beta-glucan, by definition, that can be different. And that will be differ by their specification and the purpose. But I would say beta-glucan basically once really backbone, but that has to be preserved well in terms of the length. Thank you.
Okay, thank you, Professor. So in a nutshell, the 1,3 beta-glucan probably the most effective one. Yeah, especially the pure one. Okay, then let's move on to the next one. Next one is going to be like, will too much induce immune modulation, the overstimulated, the utilize some metabolic energy in animal?
Yeah. I think I saw that question somewhere. I think that's a quite good yet scientific question. But also that has been quite important question for the field as well. There are some other similar questions. So I would say two different issues together, but related issues. First of all, so when you use the immunomodulant, I think that's quite important. When you do not use anti-inflammatory reagent, I think that's also important. I always emphasize that we have to know exactly when to use and not to use, because we know that even if it's good for the health, if you have taken too much, that's no good, and therefore that has to be balanced. For example, let's say there's a weaning condition that we know what happens at the weaning. They will have some problem with the feed, and also there is some damage, and that will be stressful condition, and therefore they will have some diarrhea, but non-infectious diarrhea. There's a stress-induced or stress-related diarrhea that further developed into infection-related diarrhea, rotavirus, for instance. And therefore, we know that they will have some problem and they will suffer from those weaning conditions. And perhaps they need better immune systems to prepare those situations. We know it's coming. And that situation, I think there's a timing issues, and you don't have to give immunomodulant, I don't know, at the end of the growing period, but certain point will be more important others. Now, another question that Eileen asked was whether if this immunomodulant is fed to animal and then they are acting to it because they recognize it, and therefore they will induce all these actions that I described in immune responses. That's true. When it comes to here, then why we do vaccination? Well, we all know Why we have insurance while you're driving your car? Well, our goal is not to have any accident at all. So to be safe. Yet we never know what's going to happen. And therefore we do have insurance. And similar situation that when you use this vaccination, when you use this immunomodulant, and perhaps that's a trade-off. Yet when you use the immunomodulant, how much energy is used for inducing those immune response, making effector molecules and effector cells, will be much less than vaccination. Now, there's another question asking whether we don't need a vaccination if we use enough immunomodulant. That's not true. And when you use the properly, then efficacy of the vaccination will be even higher. But vaccination is about specificity. Excuse me. So we have to look into different situations. I think that's enough.
Okay, so Professor, so you already touched upon all the questions related to vaccination and application of the immune modulant. So this is going to be the last question of the day. Otherwise, we cannot accommodate all the questions anyway. So this is going to be the last question. How to measure innate immunity in the field?
How to measure innate immunity? Yeah, I saw that question too. That's a good one. And I need more collaboration and interaction with the people in the field and companies for that one. But what I understood is that one hand, something is already used among vet is that acute phase proteins. Acute phase protein has something to do with stress conditions. So that can tell reasonably well about the stress conditions. But if you'd like to check innate immune response, then it will be very hard to see immune proteins in the serum after you provide this immunomodulant. What we do in the lab is that we check and compare those increase of immune response with the vaccination, because we know vaccination induces immune response for sure. And therefore, if you measure and look into condition where we have vaccination, no vaccination, but there's one study that I showed you have designed that way. It's well designed so that you have another one, vaccination, but together with immunomodulant, and then you can tell by looking at the differences between those vaccination and no vaccination and vaccination together with immunomodulant. Would that be enough, Eileen?
Yeah, the professor, so your suggestion is probably there is a way we can check acute protein in the serum, in the blood, but most commonly, maybe we can check vaccination efficacy between the control and then treated with the immune modulant.
Right, true. Acute phase protein will not be accurate because some other stress conditions will also have this immune protein production, let's say certain infection, for instance.
Okay, so all right, with that, the recap of the Q&A session, we have actually totally 33 questions at the Q&A menu. This one probably all the participants cannot see the question, but we will, don't worry, we will summarize all the questions, then answer to you by email one by one. So you don't worry if your question wasn't raised at the Q&A session, please don't be disappointed. We will get to you with a detailed answer.
Okay. Good
and then,
suggestion, Eileen. Eileen and I will work on those questions and we will summarize and categorize into few and then perhaps Eileen will get back to all those people.
Yeah, and then we won't forget there are also some more questions in the chat box, and just like as I suggested at the beginning. So we won't forget those questions you raised at the chat box as well. Don't worry.
Sure.
Okay, so with that, we try to ending the whole session. So once again, I like to invite Professor Yoon for his last comment.
Oh, me?
Yeah.
Well, be safe and healthy. And I hope that we can meet in person. Well, webinar is fine, but I think it will be much more fun to have a direct interaction among ourselves. So we may take another chance to see each other. And I would like to thank everybody for participating.
Okay, thank you, Professor. Thank you so much for joining today's Chemin Masterclass session with Professor Yoon from Seoul National University, Korea. And then stay tuned for more Chemin Masterclass coming soon, continuously every week. Thank you so much and have a great day. Bye. Bye for now.
Bye.